Hi everyone, and thank you for being here today. I'm really excited to be part of the NCBRS Foundation, and I only have Lee and Nuala to thank for inviting me to be part of this great team. I am also originally from Cyprus, similar to Paula, and we actually know each other very well. We work on several genetic syndromes together, and today I'm going to be briefly speaking to you about the genetics of NCBRS. So just briefly about myself, I'm a molecular biologist by training. I've got a PhD in genetics of neurological disorders, and I've been in the United Kingdom since 2009 when I came to do my studies, and have since graduated, been working at the Queen Square Institute of Neurology, which is, for those who don't know, the neurology hub of University College London and University College Hospital. So I'll start off with some basics about genetics, and I'll try my best to explain things in a simple and lay way. So what is a DNA? It's a molecule that contains the genetic code that is unique to every individual. Think of it as a code, as an instruction manual for making all the proteins that form our bodies and help them thrive. A series or a sequence of these bases that make our DNA form what we call a gene, and we as humans carry around 20,000 genes that are responsible for different characteristics, different cell processes or behaviors of our bodies. Each gene provides the code and the instructions to the body to make a functional form of a substance that we will call proteins. And proteins do most of the work in our cells. They are important for the structure, the function, and regulation of the body. In the case of Nicolaides-Baraitser syndrome, the gene responsible is SMARCA2 that was discovered around 13 years ago now by a team in Belgium, and together with Sergio Sousa, who was at the time based at Great Ormond Street Hospital at Queen Square in London. They published around 45 individuals with variants in SMARCA2, and the gene really provides instructions for making one piece or subunit of a group of similar protein complexes that we call SWI-SNF complexes. They reported that the gene is important for regulating a process that we call gene transcription, which in simple term means how the DNA is transcribed into another type of molecule that we call RNA, that then gets translated into a protein with a function. This gene provides instructions for making this subunit, and this SWI-SNF complex that I've mentioned regulate gene activity or gene expression by a process known as chromatin remodeling. So chromatin is a network of different DNA and protein molecules that package DNA into the chromosome, and then the structure of chromatin can be changed or remodeled to change how tightly the DNA is packed. So chromatin remodeling is one way that gene expression gets regulated during development of human bodies. And then when DNA is very tightly packed, gene expression is lower than when the DNA is loosely packed. And to provide energy for this process, the SMARCA2 protein involved in NCBRS uses a molecule that we call ATP. The SMARCA2 different mutations that cause Nicolaides-Baraitser syndrome result in the production of an altered protein that gets to interfere with the normal function of this complex. And then these altered proteins are able to form SWI-SNF complexes, but then these complexes become non-functional. And as a result, they cannot participate in this process that we call chromatin remodeling. So disturbance of this regulatory process can alter the activity of many genes, which likely explain the diverse signs and symptoms that we see in individuals presenting with NCBRS. Regarding inheritance of SMARCA2, here a term that we call de novo is used to describe a change in the DNA sequence of a gene that is seen for the first time in a person and that has not appeared in any previous generation. In other words, it is not inherited and you can't expect it or be aware of it beforehand. But this is how SMARCA2 has been found to be genetically inherited. A variant means a specific change in the SMARCA2 gene that has been found through a blood test that basically looks at many genes at once. And most pathogenic variants that are linked to NCBRS have been found in exons 15 to 25, which make up this very important ATPase domain of the gene. And these mutations usually cause a dominant negative or a gain of function effect. But what does gain of function mean? It basically means that the gene or the protein is either turned on too much or at the wrong time and place, or that the mutation results in a gain of a new or abnormal function that doesn't usually have. So this makes the protein behave in an abnormal way, which can affect how cells and the human brain then develops. So there's other similar complex genes to SMARCA2. One very relevant gene is SMARCA4, and mutations in SMARCA4 cause a very related condition that is called Coffin-Siris syndrome, but it's quite different to NCBRS. Also, it's quite rare for a second child in the same family to have NCBRS, because most of cases happen, as we said, as new or de novo mutations. Nevertheless, we have seen a rare number of families that might have multiple affected children with the same mutation, and this is quite interesting for us, and we are looking into it. So in terms of planning pregnancies, families can in fact consider prenatal testing to check for any future pregnancy if they wish. If an individual with NCBRS plans to have children, there is around a 50% chance that their child will also have NCBRS. And as always, we strongly recommend that genetic counseling is used to help families understand the risks and support with decision making. Patient registries are, as some of you may know, organized systems where we can collect consistent and detailed information about people with a specific condition over a length of time. They help doctors and researchers like us to understand how symptoms vary, to track outcomes, but also find patterns that have been linked with different mutations. And this information is very useful. It supports better diagnosis, better care, and any future development of treatments. So the registries that we are actually closely working with are CORTS. Some of you may have used it. It's a global rare disease registry that shares data for research purposes. We also use the NCBRS Worldwide Foundation Registry, which is a dedicated resource for the families and the researchers that focus on Nicolaides-Baraitser syndrome research. And we also use some other publicly available registries or websites like PubMed, where we can find publications, or also CleanVar, where we can find specific mutations related to genes that we are interested in. So we have collected detailed clinical and genetic data from around 185 patients through a combination of international collaboration, clinical case reports, and prospective recruitment. We compiled the largest cohort of individuals with NCBRS to date, and we are using a multi-pronged recruitment strategy. We have assessed both clinical and genetic data through the registry courts, publicly available repositories such as ClinVar and GeneMatcher, but as well through the NCBRS Foundation, which has facilitated patient and clinician referrals. So in total, our cohort includes 185 patients. There's a gender distribution of 60-40 male to female, which is consistent with prior observations of a slight male predominance. And for each case, we have collected detailed clinical data, neurodevelopmental milestones, seizure types, dysmorphic assessments, as well as variant level genetic data with annotation of the affected domains and the residue conservation. So variants seem to be clustered in the ATPase domain, particularly in the helicase C region, and we have identified multiple recurrent substitutions at several positions, mainly at arginine 1159, arginine 1160, arginine 1162, and arginine 1105, which are all located within the functionally conserved motifs involved within the ATPase domain and having an impact on the hydrolysis and nucleosome sliding of ATPase. Variants outside the ATPase domain are less common and seem to be more associated with the atypical or the milder phenotypes. And very importantly, we have also 5 patients with variants in the bromo domain here, which exhibit intermediary phenotypes, raising questions about a second upcoming domain-specific contribution to SMARCA2 function. So the question that comes to mind now is whether certain variants are more severe in the phenotype that they cause. And to address this, we would like to generate iPSC cell models from patients with different SMARCA2 genotypes that will include both hotspot but also non-hotspot variants. We are planning to differentiate these into neuronal progenitors and cortical neurons that will be able to tell us more about any variant-dependent changes in the chromatin accessibility and BAF complex recruitment. But the success of this work really depends heavily on continued clinical engagement and patient involvement. Many of the most important lines we are generating have come through direct collaboration with families, foundations, and clinicians who have been sharing samples and phenotypic details. And this is very important because we can then look at clarifying emerging genotype-phenotype subtypes. We would like to ideally expand the diversity of molecular models that we get to establish. And obviously, most important, to accelerate the path towards a targeted therapeutic testing. Why is it important for us? Really, our goal is to understand more about NCBRS, improve the outcomes for everybody that is affected, and especially because the condition is very rare, every single family's data helps us to build a bigger picture. And by working together, we can create the largest SMARCA2 dataset that will be able to guide researchers like me, but everybody else interested in the syndrome, and to plan care and treatment for the families. There is no clear correlation between genotype and phenotype. So right now there's no reliable way to predict symptoms based on the exact SMARCA2 mutation. People with different mutation can have similar challenges or very different ones. There's been some patterns that are emerging. For example, mutations in one part of the gene, the C-terminal helicase region of the ATPase domain, are looking to be more linked to severe intellectual disability and epilepsy, but this doesn't really apply to everybody. So DNA banking really matters here, and storing DNA sample helps to ensure access for future testing and research. And as we see science improving, this could support better care and treatment options later on. So in regards to the blood sample that the Foundation is planning for today, we are interested in isolating PBMC. So these are cells developed in the bone marrow from hematopoietic stem cells. They are present in the peripheral blood. So just with a small blood sample from you, we can isolate different types of cells. And these are important cells because we can reprogram them in a lab to revert them back to their initial state where they were pluripotent stem cells. And these kind of cells have very important properties. So they can self-renew, maintaining an undifferentiated state. And that subsequently means that we can differentiate them into any other specialized type of cell that we would like to use as a model for studying this disease. In the case of NCBRS, we are interested in differentiating them into neuronal cell lines that very importantly would be patient-derived, coming from affected individuals, and they would carry the genetic background that has predisposed your child to NCBRS. In the UK, we have done something very similar. So we have collected and processed PBMCs through a procedure that has been established in our lab at University College London and in collaboration with ECAC, which is the European Collection of Authenticated Cell Cultures. And samples were collected last year at the UK-based conference and have been cryo-stored in 2 separate locations for secure long-term use and future research. In the US, we are working very closely with the Cornell Institute, which is and has been a trusted partner in rare disease biobanking. And today, Cornell is here to support with sample collection that will help us to grow the research community and deepen any further understanding for NCBRS. In terms of future research directions that we have as a foundation, there's a series of SMARCA2 inhibitors that have been developed for applications in oncology, and we are now seeking to explore the potential for repurposing these compounds for treatment for NCBRS. And one of our undergoing and planned pilot studies is to generate resources that will be made available to researchers that are interested in studying NCBRS. So working very closely with the Worldwide Foundation, we are collecting this biomaterial, as I said, to have access to generating patient-derived stem cells that could be used for trialing these compounds. We would like to engineer model cell lines that will be model systems for testing these compounds, and we can test effectiveness of the compounds, and we can use different tools such as enzymatic activity assays or RNA sequencing and biochemical assays to assess how the BAF complexes are behaving. So going forward, we plan to expand the epigenomic profiling of patient-derived neurons. We would like to develop variant-specific BAF activity assays that we can use in these patient lines, and we would like to continue with testing small molecule modulators such as ATPase inhibitors or PROTACs to explore more targeted functional rescue. Our ultimate goal is to understand how distinct variants in SMARCA2 disrupt the chromatin remodeling process in the developing brain, and whether these effects can be selectively reversed. And one last mention is that we are working closely with the designers of an AI tool, which is called Gestalt Matcher AI, where we would like to use any facial pictures from affected individuals to investigate whether there is a trend in the frequency of facial features that we see with specific SMARCA2 variants. Thank you very much, and I'm very happy to take any questions after this presentation, or also you can contact me directly through the foundation. Thank you very much.